to identify, isolate, and follow the behavior of T cells specific for an antigen of choice. Fluorescent MHC peptide tetramers or MHC tetramers are used. In the process of generation of MHC tetramers, four identical MHC molecules, either class 1 or class 2, each containing a bound peptide, are used. In this video, each MHC class 1 molecule with their bound peptides are conjugated to biotin. After the conjugation, purified biotinylated MHC class 1 molecules with their bound peptides are collected. In this process, a fluorescently labeled streptavidin is also used. A streptavidin has 4 high affinity binding sites. Purified biotinylated MHC class 1 molecules with their bound peptides are then mixed with fluorescently labeled streptavidin. In the ideal situation, 4 purified biotinylated MHC class 1 molecules with their bound peptides bind to 4 high affinity binding sites of fluorescent streptavidin. As a result, an MHC tetramer is formed. Now, fluorescent MHC tetramer is added to a population of T cells. In that population of T cell, some T cells have CD8 receptors on their surfaces. These type of T cells are called as CD8 plus T cells. The TCRs present on the surfaces of these CD8 plus T cells are specific for the peptide MHC complexes of the MHC tetramer. So, fluorescent MHC tetramers binds only to the CD8 plus T cells with TCRs specific for the peptide MHC complexes of the MHC tetramer. Due to this binding, the subpopulation of T cells that are specific for the target antigen are fluorescently tagged. In the next step, flow cytometer and/or fluorescent microscopy readily detect these fluorescently tagged subpopulation of T cells. Now, some important points. By using MHC tetramer technology, we can determine the number, locally, and phenotype of cells in a population that have TCRs specific for that particular antigen, that is, that particular MHC peptide combination before, during, and after an infection. Flow cytometry is sensitive enough to detect antigen-specific T cells even when their frequency in the CD8 plus population is as low as 0.1%. Tetramer-based studies have been useful in tracing the phenotype and fate of autoreactive T cells. Tetramers of MHC class 2 proteins with bound peptides can identify antigen-specific CD4 plus T cells including Th and Treg cells specific for pathogen or autoantigen derived peptides. Dodecamers means 12mers can detect antigen-specific T cells with lower levels of TCR including immature T cells in the thymus and naive peripheral T cells. Please like, subscribe and share.